Hi everyone, my name is Tan Xia and with the metric number AW220239, this video was done to explain biosurfactant production by lactic acid bacteria isolated from homemade vegetables pickles. Biosurfactants are surface active compounds produced by diverse microorganisms at the microbial cell surface or excreted extracellularly. They can be divided into two basic groups, which are synthetic surfactant and biosurfactant. Synthetic surfactants are chemically synthesized through organic chemical reaction, while biosurfactants are excreted by microorganisms such as bacteria, yeast, and fungi. Biosurfactant has special advantage over synthetic surfactants such as biodegradability, pH, and salinity. Bacteria are the main group of organisms generating biosurfactant to microorganisms such as Pseudomonas, Excitobacter, Bacillus, and other various types of bacteria. Next, the objective for this experiment is to identify efficient biosurfactant producing lab strains from homemade pickled vegetables to assess the active microbial activity of biosurfactant producing probiotic lab and to investigate the probiotic and biosurfactant property of lab isolated from homemade pickled vegetables. So, to investigate the probiotic property of lab, we use pH tolerance and NaCl tolerance. Here is a video to show the result of this test.
For pH of 5, we do again have cloudiness. We have that turbidity for both S. cerevisiae and S. epidermis. And so I would say that is also positive turbidity, positive growth. This is our 7% NaCl. That just means sodium chloride. This is our 1% sodium chloride, or NaCl. You'll see it right. Nutrient broth. Then for our 7% sodium chloride, I would say that is positive turbidity, positive growth compared to our non-inoculated nutrient broth here for both S. epidermis and S. cerevisiae. And finally, we have our 1% nutrient of the sodium chloride for both S. cerevisiae and S. epidermis. And I would also conclude that there is positive turbidity, positive growth in both of those.
Based on the result, all isolated bacteria can resist the acid condition under the pH 1, 3, and 5, while for the NaCl tolerance, it managed to resist high concentration of NaCl, which is 3% and 5%. Next is to assess the antimicrobial activity of biosurfactant producing probiotic lab. Based on the result, all isolated bacteria show positive result, which inhibition zone are above 12 mm, but the best result get from experience is C3 and G6. Here is another example of the video.
After overnight incubation, examine your Mueller-Hinton plate. If zones appear around the discs, then the organism is sensitive to the antibiotic. However, if no zones appear around the discs, then the organism is resistant to the antibiotic. Sensitivity patterns can also be measured and recorded as either sensitive or resistant by measuring the inhibition zone size and comparing the results against a reference table of antibiotic zone sizes.
Lastly, to investigate the probiotic and biosurfactant property of lab isolated from homemade pickled vegetables, we use oil spreading technique. This technique is used to determine the presence of biosurfactant production. Based on the result, all isolated bacteria show the positive result, which all have biosurfactant production. An example of video will be shown. In conclusion, 8 lactic acid bacteria were successfully isolated according to the data obtained. It consists of all isolated bacteria as gram-positive bacteria. The isolated bacteria have capacity for probiotics such as low pH, salinity, and antimicrobial ability. In a recent study, the most potential producer of biosurfactant was found to be all bacteria isolate identified as lactic acid bacteria. That's all from me. Thank you.